everyone, thank you for joining for another video. Today we are looking at how drugs, including addiction and alcohol, interfere with these communications and can lead to addictions. We also explore the role of neurotransmitters in mental health and epilepsy. Don't forget to check out our previous part 1 video on how your body communicates within itself through nerve impulses and synaptic transmissions, what neurotransmitters are and their roles within the body. So today we're going to start by looking at the biological basis of drugs and the effect of drugs on neurotransmitters, followed by the effect of epilepsy and treatment, and finally drugs and addiction. So starting with the biological basis of drugs. Drugs can either facilitate or inhibit synaptic transmission between the presynaptic and postsynaptic neuron. Agnostics are drugs that mimic or increase the effects of a neurotransmitter, whilst agnostics are drugs that block or reduce the effects of a neurotransmitter. Drugs work by doing one or more of the following to neurotransmitters: increasing the synthesis of neurotransmitters within the presynaptic neuron, causing vesicles to leak in the synaptic gap, therefore increasing release, decreasing reuptake of neurotransmitter from the synapse to the presynaptic neuron, blocking the breakdown into inactive chemicals, directly stimulating or blocking postsynaptic receptors. Most of this increases the chances of the synaptic neuron being activated or stimulated, which is an agnostic response. Only those that block or destroy the neurotransmitters to be released are agnostic, which is where they're stopping the message from being transferred. So if we move to look at epilepsy, epilepsy is the tendency to have repeated seizures that start in the brain. The neurotransmitter stays within the receptor, which leads to an overstimulation of the postsynaptic neuron, and there are too many messages being sent to your brain. These spread quickly due to the neurons being connected in a network, thus seizure can spread quickly if it is not blocked. There are some brain procedures that can cause a seizure. This is from overexcitement of neurons by sending too many messages, which disrupts normal communication. A connected group of neurons sends out an abnormal excitatory message, which is larger than normal depolarization, and this abnormal message spreads quickly. Neurons might behave differently to normal because neurons are damaged and they send signals uncontrollably. There are too many or not enough neurotransmitters. Ion channels on neurons close slower than normal due to damage. If this happens to sodium channels, there is a continual action potential. And neurons might have different synaptic receptors to receive neurotransmitters. So if we look at the different types of epilepsy treatment, there are anti-epileptic drugs, or AEDs, which reduce the likelihood of seizures by altering or reducing the excessive electrical activity in postsynaptic neurons. Different AEDs work in different ways to have different effects on the brain. They either attach themselves to the neuron surface or receptors to change the activity of the neuron, or directly affect neurotransmitters responsible for sending the message. Epilepsy treatment also affects sodium ion channels. AEDs bind themselves to the sodium channels on neurotransmitters, which prevents channels from opening. This affects how ions flow into the neuron and stops the channel being activated, which prevents an action potential. This affects how fast or well sodium channels work and stops neurons sending repeated messages. Examples of this include phenytonin and lamotrigine. AEDs can also affect calcium ion channels by blocking these channels. Calcium usually enters the presynaptic neuron to help neurotransmitters be released into the synapse, without which no message is transferred. This stops the release of neurotransmitters and prevents messages being sent along the synapse to the next neuron. Examples of this include topiramate and lamotrigine. GABA, or GABA-aminobutyric acid, system and receptor agonists. AEDs can increase GABA effect on movement of chloride into neurons. This speeds up the switching off of action potentials. AEDs can increase the production of GABA, which increases levels of inhibitory neurotransmitters. Examples of this are clonazepam and phenobarbital. AEDs also have an effect on glutamate, which is a major excitatory neurotransmitter. AEDs can prevent glutamate uptake in the postsynaptic neuron, which prevents glutamate from helping movement of ions, and this prevents the spread of messages between neurons. An example of this is topiramate. Now let's look at drug effects. So the circulation of sodium is highly important. Local anaesthetics work by attaching to the sodium channels and preventing them from opening. This means that pain is detected by the sensory neurons, but this pain signal does not travel to the brain and therefore we do not feel it. Scorpion venom works by attaching to the sodium channels and preventing them from closing, so the opposite effect. This allows sodium to build up in the neuron and eventually become toxic. Now let's look at addiction from drug use and recreational drugs. Within this, there is the effects of dopamine and mental health. Too much dopamine is what we liken to positive symptoms of mental health. So this includes hallucinations and paranoia as part of schizophrenia, uncontrolled speech and movement as part of Tourette's, agitation and repetitive action as part of OCD, and overexcitement and euphoria as part of mania. When you have too little dopamine, this can present as negative symptoms, which can include tremor and inability to start movement within PD, feelings of lethargy and misery within depression, lack of attention and concentration within ADHD, and cravings and withdrawal. Dopamine is an excitatory hormone. Addictive substances increase dopamine activity in certain areas of the brain, the reward pathway. This can often be seen through sexual intercourse or gambling, which create a feeling of pleasure and happiness. Olds and Milner in 1954 placed rats in a Skinner box that allowed self-stimulation of the brain by the pressing of a lever, which triggered their reward system. The rats learned the association between pressing the lever and stimulating the reward system due to the release of dopamine. The rats sometimes pressed the lever 2,000 times per hour to stimulate the release of dopamine, as the rats had developed an addiction. Other behaviours that release dopamine include sexual excitement, gambling, and video games. fMRI research indicates dopamine is released during viewing of attractive people as well. Almost all abused drugs stimulate dopamine release in the nucleus accumbens. This is a small subcortical area rich in dopamine receptors, and it's the area responsible for feelings of pleasure. Sustained bursts of dopamine in the nucleus accumbens inhibit cells that release the inhibitory neurotransmitter GABA. Stimulant drugs increase excitement, alertness, motor activity, and elevate mood due to inhibiting the inhibitory system reduced GABA. Some examples of this are amphetamine, which stimulates dopamine synapses by increasing the release of dopamine from the presynaptic terminal. Cocaine, which blocks the reuptake of dopamine, norepinephrine, and serotonin. The receptors stay in the synaptic gap and are much more likely to stimulate the receptors again. Ritalin, or as it's otherwise known, methylphenidate, also blocks the reuptake of dopamine, but in a more gradual and more controlled rate, and this is often prescribed for people who have ADHD. So let's move on to alcohol. Alcohol is a drug that has long historical use and is used widely throughout the world. In moderate amounts, alcohol is associated with relaxation. In greater amounts, it impairs judgment and damages the liver and other organs. Alcoholism or alcohol dependence is the continued use of alcohol despite medical or social harm, even after one has decided to quit or decrease drinking. Alcohol has a number of diverse physiological effects, including the inhibition of sodium across the membrane, expansion of the surface of membranes, decreased serotonin activity. Serotonin is the neurotransmitter responsible for uplifted mood, therefore alcohol can lower your mood. Enhanced response by the GABA receptor. This means the postsynaptic neuron will be even more inhibited as GABA will inhibit it. Blockage of glutamate receptors, which would prevent the postsynaptic neuron from being activated, and increased dopamine activity, which can lead to hallucinations, paranoia, etc. Alcohol is inhibitory. It inhibits the center of inhibition. With less inhibition, we can be more social, although too much alcohol can enhance sad feelings, leading to depression. Addiction has various factors contributing to continued substance abuse. Tolerance develops. Therefore, individuals feel they need more of the substance to receive the same effect on the reward system. There is a back and forth between pleasure from the drug decreasing and withdrawal symptoms, which are very unpleasant side effects, which lead to taking more of the drug after a shorter period of time between doses. There are also cravings in response to cues from the environment. These cues trigger cravings. Even if you are not addicted to alcohol, seeing an advert for alcohol can make you crave it. Those with an addiction would really feel a need to get more. And brain reorganization. The nucleus accumbens and prefrontal cortex. The brain structures can be altered. Whilst looking at addiction, it's really interesting to also consider increased attentional biases. So attention is biased towards particular stimuli that are relevant to us. Attentional biases can even predict heroin relapse. Marissen et al. In 2006 wanted to examine whether attentional biases could even predict heroin relapse. This was done through an emotional Stroop task where participants were presented with words, for example, the word green, but the colour does not match the word itself. For example, the word green is coloured red. The participants are asked to name the colour and not the word. The participant must focus on the colour and ignore the word. The time that they take to answer is measured, and the level of inference the word has upon the task. Every so often, the word will be relevant to the type of addiction the participant may be suffering. For example, dealer. When the word is strongly associated to a person's addiction, it will take them longer to identify the colour of the word due to the word interfering with the participant's concentration. Pre-treatment attentional biases can even predict relapse at a 3-month follow-up. Those that were showing a strong attentional bias, so they took longer to name the colour for addiction-based words towards the words relevant to their addiction, were also the people who relapsed 3 months during a follow-up later. The strength of attentional bias could predict likelihood of relapse and could become an instrument that's used to do so. However, other research shows that even social drinkers show different attentional biases to alcohol. This was investigated by Townshend and Duka in 2001 through a dot probe task. Participants were presented with a cross in the middle of the screen and told to look at it. They were shown a picture and have to identify whether the dot is shown to the left or the right of the cross. They measured the time taken to identify the dot. If an alcohol picture is also presented before the dot is shown, for example, the alcohol picture is on the right and then the dot appears on the left as the alcohol picture is removed, the participant will take longer to identify the dot due to their attention being distracted by the alcohol. Their attention has shifted to the right of the screen and it takes longer to shift it back to the left. This can be controlled by also presenting neutral stimuli, which did not affect the attention or time taken to notice the dot. Interestingly, if we also look at occasional drinkers, where they would actively avoid bringing their attention to the alcohol picture, which produced a negative bias score, whereas heavy drinkers show a really strong approach for a positive score as they take longer to shift their attention from the side where the alcohol picture used to be to where the new dot has formed. Finally, let's finish on how we can prevent addiction. There are several medications that are used to combat addictions, and these include antabuse and methadone. And these are used as many do not respond to other treatments, so medications have been used to reduce cravings that one may experience. I hope you found this video interesting, and I look forward to sharing more with you soon. Thanks, bye.